Hi all, welcome back to Microbiology Lectures, and today we're going to continue talking about fungi. Last time we talked about the makeup of their cell wall, and today we're going to talk about structure and then classification. Okay, so fungi themselves can be both, well, they can exist in multiple kind of morphologies. Remember, we talked about three main morphologies. You have a yeast, a mold, and a mushroom. And so lots of fungi actually can be dimorphic in the sense that depending on the environment in which they're raised in, they might be in a mold form or a yeast form, and they might transition between the two. They could even be in a mold form and then transition into a mushroom form when conditions get right, so they can produce spores. And so you have these spores, and if they're single spored and live kind of as a single cell, we classify them, their morphological type is yeast. When they're multi-celled and the cells are either connected or not connected by septa, but when they occur in long strands, multiple cells together, they're called hyphae. And you have a bunch of hyphae that are connected. It's called mycelium. And so again, most of you've probably seen mycelium before. If you've seen bread mold or really mold on anything, you'll see these little hair-like structures. Those are the hyphae, and then collectively the mass is called the mycelium. But when I was talking about the division, okay, so in septate hyphae they have septa, which separates each cell, and there are really two classifications. Most species are septate hyphae, but some are not. Some are actually synodic hyphae, and so there's no septa in those, and the cytoplasm is completely connected throughout the entire hyphae. And so here you can see the septate hyphae. They have these little septa or septum that separate two nuclei or two cells, except for they're porous, so they actually have holes in them, and that allows the cytoplasm to kind of be connected, so they can exchange nutrients between cells very easily. And so you might have part of the hyphae that is digesting material, and another part that's growing in a separate direction, potentially even reproducing, and they can share nutrients that way. In the case of conidic, it's just all connected, and like there's sharing at all times. So anyhow, those are really the two types of hyphae that occur in the fungi kingdom. There's probably other variations of it, which we're not really going to talk about. Okay, so most people know that fungi are not motile. They don't move, except for they can grow towards a food source or a nutrient source. And so much like a plant that's rooted in the ground and its roots grow towards a water source or a nutrient source, fungi are similar in the sense that the hyphae will travel towards a nutrient source, and the hyphae portions that are kind of traveling towards that nutrient source and have the ability to digest it and excrete enzymes to do so, we normally call those vegetative hyphae. And then they will produce hyphae that are typically encountering the air, so they're above the material. So this could be above the ground. This could be above the flesh of an organism. But those are aerial hyphae, and they typically are the ones that are going to produce sexual or reproducing cells. It doesn't have to be sexual. It could be asexual reproducing cells, spores in general. Okay, and sometimes we classify species of fungi based on their aerial hyphae. So just by looking at the aerial hyphae, we can say, oh, well, this is the color and the structure and kind of how they connect, how the mycelium looks. It gives us an idea of what species that might be, or at least what group it might fall into. So here's kind of just a diagram of that. The vegetative hyphae is typically the hyphae that you really don't get to see. Maybe, this is the surface of the soil, this is the surface of the skin, something. The vegetative hyphae is the hyphae that's going to be digesting material underneath, and then the aerial hyphae is the stuff that's going to be most of the time reproductively active. Okay, so it's producing spores. It's ready to allow those spores to either be carried by the wind or water by another organism, something that's going to pick up that spore and take it somewhere else. And so the whole thing is a thallus, or the whole thing is a mycelium. But we divide the two mainly to talk about, well, the reproductive part, or the aerial hyphae, and then the vegetative part, or the part that's actually consuming material. Okay, so like I said before, they can do asexual reproduction or sexual reproduction, or both. And you can actually, this is an interesting piece, you can have hyphae that in one part of the hyphae, they're producing a mushroom that's going through sexual reproduction, and then the other part of the hyphae, they're going through asexual reproduction and just growing towards another food source. And so you can have the organism itself going through asexual reproduction and sexual reproduction at the same time. And so asexual reproduction, it really depends on the species. This is where we get into a lot of the terminology that is species or group dependent, and that will be based on like what type of spore do they form. And so like, is it a spore that is formed from conidia, and if it's a conidia form or conidia spores, then they belong to that group. Is it a sporangium or a sporangiospore that is encapsulated here? That they belong to that group. Is the asexual reproduction not a spore that's typically carried by wind or water, but rather maybe fragmented off and broken? And so that's the anthroconidia, is typically looks like hyphae, and it's just the hyphae splits, like the cell just breaks and can be carried away. Sometimes it is carried away by animals, or sometimes it's carried away by wind and water, but it's not forming these spore-like structures that are different. One key thing to remember when often people are talking about fungi, they'll say the term spore. Spore just really, it refers to reproductive cells, and these reproductive cells can be the result of asexual reproduction. Okay, so it can be mitosis, or sexual reproduction, meiosis, and so that sometimes gets confusing for students, but the term spore just means reproductive cell. Okay, and again, how these are carried, how are these distributed across the landscape really depends on the species. Again, a lot of the spores themselves are easily carried by wind and water, or even on the fur of organisms or the feathers of birds that might come in contact with the fungus. Some of them also can be digested. Quite a few can be digested. So when a mushroom forms, the spores are often held underneath in the gills, sometimes not. Sometimes it's inside the mushroom itself. If it's consumed, a lot of those spores, they have a high degree of chitin around them, and most organisms cannot digest chitin, and so that spore itself can't be digested. It's defecated somewhere else, and that mushroom can take foot or produce a hyphae in that new region. The spores themselves, a lot of times, they can persist for a very long time. We found fungal spores from ancient lake beds that still would have been a somewhat aquatic fungi. We found fungal spores inside Egyptian ruins. We found fungal spores in many places that, if given the right conditions, they germinate and produce hyphae. And so sometimes this is of concern, especially for archaeologists, individuals that are going into some of these old tombs and things. Just as we talked about the endospores for bacteria having the capability of persisting for a very long time, fungal spores can also persist for a long period of time. And you really don't know what that individual in that tomb died from. So it is potential that it could be a fungal infection in the lungs or something like that, or potentially, a bacterial infection or something like that. And they went into in the case of bacteria, an endospore form. In the case of fungi, maybe a spore form, and they're just residing in that form until conditions are right again. And so this is of concern for scientists sometimes when we're dealing with this. All right, another way that is still asexual but a little bit different is the process of budding. And so yeast can go through an asexual production that we call budding, and it's a little bit different than the mitosis, divide cells completely in half and have two daughter cells. Instead, what you have is a parent cell with a smaller daughter cell that's generated from it. The nucleus and the nuclear material is all the result of mitosis. Okay, so it's all asexual reproduction, but the budding process itself is a little bit different. So you'll have this nucleus that's going through mitosis. Okay, so if you remember back to like a general biology, mitosis is nuclear division. Okay, so we often think of mitosis itself as cell division, but that's not true. Mitosis is nuclear division. So the mitosis that happens in asexual reproduction, the formation of spores in fungi, and the mitosis that happens in asexual production in, say, like budding process, it's the same mitosis. Okay? The process then differs by what's done afterwards. And so in the case of the budding process is once the nucleus has divided and finished mitosis, one part of the nucleus travels along microtubules to a budding region of the yeast. And when the nucleus gets into the new kind of bud, then that pinches off, the bud breaks off, and you have what we typically call a parent cell and then like a smaller daughter cell. And this is just another way at which these organisms can go through asexual reproduction. Sexual reproduction in fungi It's kind of bizarre to think about, but there are two kind of mating types. We don't call them male and female, but you can kind of think of them as male and female in the sense that one is considered a plus and the other one is considered a minus. And so the hyphae itself would be considered a plus hyphae or a minus hyphae. And if a plus and minus hyphae come together, then they can start going through the germination meiosis process and produce a spore or spores that are the result of meiosis, not mitosis. And so they would not be genetically identical, so they would go through actual sexual reproduction. And so it's kind of a neat system. So here you can see you can have a species that's going through asexual reproduction, mitosis over and over and over again, still producing spores, right? And the spores look very, very similar to each other. In fact, sometimes you cannot tell the difference at all. But then conditions get right, conditions get good, maybe it's the right humidity, right temperature, right amount of nutrients, and the hyphae might come in contact with each other, a plus and a minus. When the plus and the minus come in contact with each other, fertilization occurs, and then meiosis happens. So then you go from a haploid organism to a diploid organism, and then that diploid organism would produce a fruiting body, which has spores on it, and then the spores can be released, and you have these spores, which are the result of meiosis, which are genetically different from each other, and then these spores, which are the result of mitosis, which are genetically identical to each other. So kind of a neat system, allows fungi to really adapt to lots of different environments when conditions are good, or in some cases when conditions are really bad. Maybe they go through sexual reproduction. It's species dependent, and in fact sometimes, not sometimes, there are quite a few fungi that we know about this process. We know their asexual reproduction, and we've classified it, and we know how it goes about, but we've never seen the sexual reproduction. And so with the kind of discovery of DNA, and as we progress and we know more about DNA and we know how to sequence organisms, and we start learning about kind of the sexual and asexual reproduction of fungi, we've now been able to go back and look at some things that we classified as, this is one type of mushroom and this is another type of hyphae, and we didn't put the two and two together that they were the same species until we looked at the DNA and found out, oh look, this is the hyphae of this mushroom, and this mushroom is just the sexual reproduction of this hyphae. And so there's situations where we've basically discovered things twice, and that's kind of what DNA sequencing has really gave to the study of fungi. Okay, so real quick, let's go through some classification of the different fungi. Again, they're not classified on their morphology types, so not yeast, mold, and mushroom, but rather typically they're classified based on some encapsulation of their spores. A lot of times that's going to be what we classify them on, but really classification, you could say, is up for debate, with things being genetically sequenced now and the genomes being sequenced. We're starting to recognize different species, and even things that we once classified as the same species, we're now finding out are not the same species. And so estimates right now are about 1.5 million species in nature, although we're far from that with genomes. But we do break the kingdom fungi up sometimes. Okay, so again, this is in flux. This meaning it's debated. We do often, I should say, break it up into two separate categories. The Oomycota, which are the mycelium asepta, so no septa forming, and then the Eumycota, which is the true, which means true mycota, which means their mycelium is septate. Okay, and so we think of kind of the Oomycota as the lower true fungi, maybe the first group to kind of evolve, and then the Eumycota or the true mycota as like the higher fungi, and kind of maybe a little bit more advanced structures when it comes to fungi. So mycetes are the sac fungi. These guys are fairly important for lots of reasons, but they produce a acis or an ascospore. So this acis is a sac that kind of engulfs the spore, or the spore is held in. So you can see it here. So you have these sacs that hold the spore within it, right? And when I say that they're important, it's because they're the most abundant fungi that we know of. Like lots of fungi belong in the sac fungi grouping, and from a microbiology perspective, they're extremely important because this is where penicillin lies. So it's classified in here. But other things like morels and truffles are all part of this group, and there are other things like diseases that are pathogens and things like that are in this group. So again, 75% of the recognized species fall into this group. So you can imagine that a lot of species that are important to us fit under sac fungi. The club fungi or basidomycetes, they form a basidospore. So again, like we're talking about the spore, or at least what encases the spore. So they form a club-like spore or structure, basidium, and this is the group often when people talk about mushrooms. This is what people think of when they think of a mushroom, like a mushroom cap. And so the spores are typically in a gill form underneath the cap of the mushroom, and can be distributed either as some organism comes and eats this if it's edible, or the wind blows past it. Sometimes water will run down it, and the spore might travel in water. So there are lots of ways in which the spores can travel. Lots of them are grown for food, and then lots of them are toxic. So there are quite a few that form a pernicious toxin, so a toxin that doesn't allow the proteins to form, so the amino acids to bond. And so that toxin can be very dangerous to humans. Again, pretty populated, pretty species-rich group. Some very important for things like wheat and rye, in the sense that they cause pathogens, which we call rust, in those groups. Microbiota, this is another group. They produce zygospores. They used to be called the zygomycetes, but now they're called the mycorrhizas, and they hold their spores in a zygosporangium. So we already looked at this specialized structure before, and really it's kind of interesting because they have this plus minus system also, like the species do when they do go through sexual reproduction, but they form this zygosporangium that then forms on a stalk, and the spores are held within that. So normally we see some kind of interesting structures in this, but like black mold is an example of this, and so sometimes, or black bread mold, sorry, sometimes it plays an important role in its ability to germinate and form spores on food that is important for humans. And I think many people have probably seen the actual sporangia forming at the top of like bread mold. If you look at it close, that's why it kind of gives it that black bead or black dots on the top of mold. So it's also can be important from like a food transfer perspective. Then there's the pathogenic fungi. Okay, so let's go through these fast also. Superficial fungi, so these are the ones that occur on the skin, nails, hair, that kind of stuff, keratinized tissue, and they're called dermatophytes. And so things like athlete's foot, jock itch, ringworm. So if you're an athlete or a wrestler, or basketball players, football players, hockey players, sometimes you'll get these fungi infections, which are the result of dermatophytes. There's other people who garden a lot. People are landscapers. Sometimes they can pick up what's called a rose gardener's disease, or shrinky, which is a spore trichus that can, you can just get this. It's typically inhabits soil or plant tissue that is kind of wet, and you can get a subcutaneous fungi, so one that occurs underneath the skin, and starts consuming material underneath the skin. So superficial fungi on the outside, subcutaneous on the inside, and then you have a whole bunch of others that are just opportunistic, in the sense that it just means the condition's right. So Candida albigans, females might be familiar with this. Okay, this is a yeast infection. It's just it just depends on the conditions that people find themselves in. The histoplasma, people get this from chicken fecal material. So people that work in large chicken facilities and don't wear respirators, sometimes they'll get fungus infections that occur in their lungs, that form a histoplasma, complications from that, and so there's all kinds of pathogenic fungi that occur. But again, a lot of these are opportunistic, or a lot of these occur only when conditions are perfect. So it's not like a lot of them are not transferred between individuals, although there are some, like some superficial fungi, ringworm and things like that occur on the epidermis of people that can be transferred that way. But a lot of these are not transferred from human to human contact, and so like we treat them for sure, but we don't talk much about outbreaks. And then, of course, there's the beneficial bacteria associations with like mycorrhizal. When we look at the mycorrhizal and the ability for fungus to associate with plants, we owe a lot to this relationship. In fact, we grow massive amounts of food due to this relationship. And without this relationship, we know that the plants don't grow as well. The fungi often will supply the needed nutrients that plants just can't get, the nitrogen, the phosphorus that plants can't get. And either we have mycorrhizal associations or we dump a bunch of fertilizers on a field, one or the other. And so I think at least in modern times, more people are going with the mycorrhizal to inoculate mycorrhizal into the soil system. And so you can do this many different ways, but often people are interested in that, mainly for the benefits. Like when we look at native plants and when we look at wild plants, almost 80% of all the plants out there will have some kind of mycorrhizal association. When we sterilize fields and we create a monoculture, we tend to not allow for that to happen. And so often that's problematic when it comes to that. And some plants, they just require it, like they can't live without it. And so when we look at a chart, majority of plants all have, like if we're taking plant size into account, majority of plants all benefit from mycorrhizal association. Okay, some plants can play catch up. So if you plant them both at the same time, no mycorrhizal on this dotted line, mycorrhizal on the solid line. Eventually, if enough time goes by, they can play catch up. But some, there's no catch up. So things like orchids, if you don't have that mycorrhizal association, that plant can't grow and it dies. And so some are permanently stunted because there's no mycorrhizal, and some can play catch up. It just takes them longer to grow. So we're starting to learn more about this association and the importance of the mycorrhizal or the fungi community in the plants that we grow for food, ornaments, etc. Okay. Next time we come back, we'll talk about protozoans.